Hello everyone. So today we are going to talk about the topic selection and evolution. This comes under topic 17 of 9700, which is the A level biology curriculum. It's usually towards the end of the A level biology curriculum and usually is covered in paper 4 in form of questions and under the planning paper also in paper 5. So we move on to the first topic, which comes under selection and evolution, and the first one is the variation topic. So obviously, the word variation in itself means something which is different from what is actually existent. So variations can be of three types: genetic variation, that is the one which occurs in the genes itself; phenotypic variations, which are visible on the outside. Phenotype refers to something which is visible, the physical appearance. So phenotype relates to physical here, and the environmental variation, the variation which is caused due to changes which occur in the environment in which a particular organism or a group of individuals or species are living in. When we come to talk about genetic variation, as I clearly pointed out, occurs in the genes. So when it is occurring in the genes, it has to occur because of certain changes or steps which occur during the genetic processes of meiosis or during the division process. So when the question comes in for genetic variation, they usually ask you what genetic variation is. You have to suggest what this term means and the reasons behind it. So the reasons behind it are something which we already studied during the inheritance chapter: independent assortment. Independent assortment was when each of the alleles independently separate out. Random fusion, multiple gametes are produced during the process of meiosis. Which gamete of the mother fuses with what gamete of the father is a random process. Mutations, which can be good mutations, bad mutations. These mutations are changes in the genetic sequences which will also lead to some form of genetic variation. And crossing over, which would lead to creation of completely new set of chromatids because of let's say exchange of fragments containing a certain set of genes, is what we are talking about in crossing over. The next part, which is a result of genetic variation. So if there are changes in the gene setup, there will be changes in the outcome. We've always studied that it all starts from the central process of DNA to RNA to protein. Okay, and when we are talking about this, if there are changes which occur at the DNA level, there will be changes which will eventually show up at the protein level. So this is what we refer to as phenotypic variation. As I mentioned, changes which are there on the outside or which are visible. Environmental variation, which is the last form of variation which we are talking about. In case of environmental variation, the major thing which comes in is changes on the outside which are occurring because of you trying to adapt, or an organism trying to adapt to changes which are occurring. First, we move on to the topic of genetic variation. As I mentioned, it is occurring in the genes already. They are the ones which will be affecting the gene sequence of a particular individuals. And the most common answer of genetic variation and the reason behind it is mutation. There are a few pointers which had been elaborated in the books which have been prescribed to you under Cambridge. And mutation can occur in a single cell, and mutation can occur at the same point in multiple cells. So first we come to changes in one body cell will not create such a big effect because our bodies are made up of about trillion cells. One cell and the other ones not changing that much, and that one not dividing, that mutated cell not dividing that much, will not create such a big problem. Mutation usually is visible when it occurs in multiple cells. Obviously, as I pointed out, independent assortment, crossing over, random fusion are one of the reasons for genetic variation. So, sexual reproduction is an important part of it. Mutations which are caused as a result of environment, because an individual is trying to survive in that particular environment, it's not passed on from the parent to the offspring. It is just that adjustment of that generation which occurs. So the next topic about variation, on which you're questioned a lot during your assessments, your summative, formatives, wherever you are giving, are continuous and discontinuous variation. What you need to remember for continuous variation is that continuous variation is a continuous process we are referring to, and as a result of which it will have multiple values to it. So a very common thing which is asked and is expected for you in the answer, that there will be multiple values. Because we are talking about multiple values, there will be many intermediates, and it will be helping us to measure a lot of quantitative measurements. As a result of multiple values, multiple intermediates, it will have multiple phenotypes, and the best example of continuous variations are height, weight, where you get a lot of values, and the range of values are enormous. Obviously, when we come to continuous variation, the kind of questions which are asked to you are usually sometimes in form of a histogram, where they are trying to show you a histogram where you have bars which range all across from smaller values to higher values, giving you like a curve. So please remember, continuous variation will have multiple values. Multiple values goes down to the genetics of multiple genes. If there are multiple genes, there will be multiple phenotypes. Whereas when we come to discontinuous variations have fixed values. For example, the blood group, you don't have A, B, C, D, A1, A2, A3 as blood groups. Rather, the blood groups are A, B, and O. So there are only certain values which are fixed. There are no intermediates. It does not allow quantitative measurement but provides for qualitative measurement. Furthermore, if you delve deeper into each one of these variations, so discontinuous variation, if you come to the genetic level, this is something you have to remember and has to be there in your answers when you are trying to determine the genetic effect of these variations. So there are different alleles at single locus which will have large effects. So here we are talking about one particular gene, okay, at a particular area. So the single locus is a particular area we are referring to, and many alleles in these genes will have a large effect. So if my A blood group gene is present in double quantity, it will be A positive, and both of the alleles for the blood group A will be dominant. So I have different alleles, but they are all present at the same locus and have the large effect. However, in continuous variation, we said that there are multiple genes having an effect when we were starting to explain it. So here, different alleles have very less effect at a single gene locus, but there are different genes which are present here. So that is why this type of variation comes under polygenic inheritance. The questions which are asked are explain the reasons for the kind of variation that is observed in this particular question. That means the reasons behind it are many genes. It is polygenetic, okay, where many genes have a combined effect. A very common keyword which is expected in most of the answers is additive effect of genes is 1 mark, polygenes second mark. So before we move on to the next topic, which is that of natural selection, I just want to summarize things we have dealt till now. This is where we make the transition and we start one of the starting process of this chapter, that is the selection process, But before the selection, we had introduced terms of variation. We talked about genetic variation, we talked about phenotypic variation and environmental variation. The other set of questions which come in, we talked about discontinuous variations as well as continuous variations. The next topic which we move on to, natural selection, which was introduced by Darwin, has been in your course book since the time you were all in middle school years. From about year 5 or 6, this was introduced. So natural selection in itself has a number of steps, but what you need to understand with respect to what is expected of you to answer certain set of questions, the first one comes in natural selection, where you already have certain characteristics and your particular population have certain different alleles, and those certain different alleles will be selected by the nature depending upon the changes which will occur. This might sound very confusing right now, but as I move ahead, it should be very clear. First thing, you need to understand these two terms, biotic and abiotic factors. Natural selection is affected by what factors? Biotic. Biotic refers to the living factors, that is all the plants, animals, insects, microbes living in that area. Whereas the abiotic factors which we are talking about here are the non-living factors, the wind, humidity. We are talking about the rain which is happening in that area, light intensity, changes in temperature, things which are non-living. When we move on to natural selection, the kind of questions which come to you, you are expected to answer those questions using terms such as biotic factors, abiotic factors as one set. The other set is using terms like fitness and selection pressure. So remember, when you are answering questions for selection and evolution, plan to use terms such as fitness and selection pressure. Now, what do these mean? So as I mentioned, when we are talking about natural selection, important terms such as fitness, selection pressure come into play a lot. And when we are talking about fitness, fitness refers to extent to which an organism gets adapted to the environment. So let's say in a particular setup, in a particular species, there are many organisms. The temperature drops in that certain area, as a result of which certain organisms might be able to adapt to it, certain might not be able to adapt to it. The one which are not being able to adapt to it have lower fitness as compared to the ones which have higher fitness. So whenever we are trying to write our answers, we understand what the term fitness does mean. But when we are trying to answer, rather than using better adaptability, we can try and use the term fitness, which is a better term, a better key term, trying to express that an individual can survive because of variations, because of how the fitness is. The second topic which comes in is selection pressure. So selection pressure in itself tells something which is causing selection to occur. So basically, the environmental factor which will put the pressure to select certain individuals and allow the survival of only certain organism. It can be environment, it can be presence of a particular predator which suddenly becomes very dominant in that area, and as a result of which it can cause a change in a particular set of organisms. If you remember, a very common example of industrial melanism, which has been occurring for a very long time, has been taught to you with two different type of colored moths which have existed. There the factor was completely external. Industrialization acted as a selection pressure for a particular species of moth to be dominant at a particular point of time. Steps of natural selection which are known to us, and you need to remember the order. There is certain variation which does exist in the species. This variation is passed on to the offspring through the process of inheritance, Because this offspring has it in that species, in that particular community, the selection pressure comes into play, and the ones which can survive with that variation are selected. Over the time, these divide, and as they divide, there is always survival for the fittest, because there is always a fight for food which starts to happen, and that's when adaptability comes into place. So these are the 5 steps which are involved in the steps for natural selection. And the graph which you actually see on the side is how over the time the number of populations can increase. In the starting, the selection pressure is high, only a few organisms survive, but over the time these values tend to increase, and this is what this graph is simply showing, what happens over a particular time span during the process of natural selection. When we talked about natural selection, those were the steps involved. Coming to the types of selection, and it gets divided into 3 different types depending upon what is the driving force behind these selections. The first one, which is called as the stabilizing selection. So when I'm talking about stabilizing selection, what does natural selection do? Okay, there will be one type which has existed all the time in a particular population, certain selection pressure or a factor is applied to it, and it triggers the formation of another set of organism. Now, stabilizing selection refers to when I have 2 extreme phenotypes and a middle phenotype, and it is my intermediate or middle phenotype which has been selected for survival in the particular environment. So one of the very common examples is that of the eggs for most of the organisms. So very less eggs can lead to non-survival of the species when they are produced. A very large number of eggs produced can eventually lead to death because of excessive competition. However, if I have an intermediate number, the intermediate number is neither very less and neither it is too high for competition. So a type of selection which favors the existence of the intermediate, that is neither, in this graph if you see very carefully, I have small animals and I have large animals, but my selection, the selection pressure which has been put in a way that the dominant surviving species will be the one which has an intermediate mass. So the intermediate phenotype, when is supported in a particular selection, is called as stabilizing. The next we come to is directional. Directional is one of the most easiest forms of selection, and whenever we talk about natural selection, these are the ones which we have mostly studied, where only one particular phenotype, especially the dominant one, is encouraged to survive. Because when changes in the selection pressure happen, here for example if you see in the graph, selection against very small animals, so the factor which has triggered, or the selection pressure when applied, the kind of selection has promoted the existence of only one extreme phenotype. It can only be the larger ones, it can only be the smaller ones. And as I had talked about industrial melanism, which has existed for a very long time, only one of the phenotype could exist, either that of the peppered moth or the other one. So either one of them could have existed. So the examples related to directional are industrial melanism, antibiotic resistance, where only a particular kind of microbes which are resistant to that particular antibiotic are able to survive. So the next type of selection, the third one which we talk about, is the disruptive selection. So when we talk about the disruptive selection, this is the most confusing selection. So in this case, when the selection pressure is put, so when the selection pressure is put, it does not favor the intermediate phenotypes at all. It's only the extreme phenotypes which are actually preferred. Polymorphism does exist in this population because I want the smaller organisms also and I want the larger organisms also. I have both of them. So I have body mass of both sets of organism, but I don't have the ones which are the middle levels. So the selection pressure here does not favor the existence of the intermediate type, and that is what has to be understood. The reason behind that might be the extremes in the environment which do exist, which are only supporting the two extremes of the population which can survive. So before we move on to the next topic, the most important is to summarize what we've understood till now. And at this point, the natural selection in itself, what does it mean? The steps of natural selection, as well as the three types of selections which are induced as a result of selection pressure. The first one being stabilizing, second directional, and the third disruptive. Usually questions which come in ask you the reasons behind them, ask you to interpret the graph which has been given to you and figure out which kind of selection is that, and you have to elaborate on why do you think it has happened. So your answers either relate to presence of an intermediate type, absence of intermediate type, presence of the extremes of one population over the other. But please remember, whenever you are answering these type of questions, make sure you use terms such as selection pressure and fitness while putting them through the answers. The next topic which we move on to are the basic terms, which are the first one coming into play here is genetic drift. So genetic drift is drifting from the actual genetic setup in a particular population. And here we move on to talking about changes in allele frequency that are occurring by chance, and they are not as a result of natural selection. So there is no selection pressure that I am putting in here as a result of genetic drift, but by chance there is a change in population which is occurring because of certain population, let's say, moving from one part to another, and that kind of triggering the entire process. So it is not a process which relates directly to natural selection. Gene pool is one of the terms which is used while explaining the genetic drift. So gene pool, please remember very very carefully that whenever we are talking about gene pool, we are talking about the complete set of genetic information or all the genes of different species which are present in a particular population. When we talk about genetic drift and when we talk about gene pool, there are certain number of effects which come into place, and the reasons these genetic drift are occurring, they lead to existence of certain effect. The first one which you need to know is called as the founder effect. Okay, the founder effect is that there is reduction in the gene pool. So you can read it here, I've written it for you. It is reduction in the gene pool in comparison with the whole population, resulting from 2 or 3 individuals. So let's say in a particular population it has about different type of individuals which exist of different colors. Due to the movement of one or two individuals in that particular area towards another direction, just by chance, not because of presence of food, temperature, just by chance that they tend to move there, they are found to be dominant in a particular area in the same region as compared to the other colors which are dominant in the other area. So I'll give you a very common. So the example I was trying to talk about here, which is found in most of the Cambridge books and is very much existent in most of the study material which is provided to explain this effect, is the founder effect, is the anole lizards. So the anole lizards which are there are usually found in the Caribbean, and they are of different colors. And when we are talking about these lizards, it starts with an A, and they're called as the anole lizards. And these lizards can be of green color, brown color, they can have a checkered appearance, some of them are a little bit dark gray in color, and they exist in large numbers. So, what happens in this process is that at some point of time, let's say they are these green and the brown lizards, which while near the shore climb onto certain rocks, and these rocks tend to float away, as a result of which they move on to another part of the island and they start to grow and divide there. It is not that a particular selection pressure has triggered the existence of these, but it is just by chance that some of them have moved on to another part of the island and started to divide. So, as a result of this, what you have to remember in this is that the gene pool of these entire lizard remains constant, because there is no death of particular set of lizards, or let's say they are not able to survive because of certain selection pressure. They're all surviving, it's just that they are moving on to another area, and the area initially which had all the lizards now only has, let's say, only the black ones or the dark gray ones which are there, and the others have moved on to nearby area. But yes, they have not become extinct in number, their gene pool has not reduced completely. It's just that now I can see two different set of numbers existing on the same island rather than all of them existing together at the same point, and this all happens by chance. So when we are talking about founder's effect, please remember there are certain terms which you should be using. Talking about by chance, it's happening by chance, the gene pool remains constant, there is no extinction which is happening. And very important, before we move on to the other effect, and as we move on to the other slide, the next effect after the founder effect which we come into study during this process is the bottleneck effect. The bottleneck effect is when there is a reduction in the entire gene pool. So when we were talking about the founder effect, the entire gene pool remains the same. So I have a lot of green, I have a lot of blue, I have a lot of brown, but it's just that they are scattered differently in the same area. Some are dominant in one particular areas and the other ones are dominant in the other area. However, when we come to the bottleneck effect, it does point out that there is a reduction in the entire gene pool. So this is a very common diagram which is found in most of the books, most of the online research videos, a very common diagram which is found. So if you see, the first bottle has a lot of yellow, green and red, but the last one which the population is created, you see there is a lot of red. So the actual gene pool has reduced. And why does it get reduced? So the bottleneck event that occurs, or a particular event has suddenly led to the loss of a particular kind of the species in a particular area. For example, very common example are that of certain species which become extinct. For example, when we talk about the white tiger, the Bengal tigers, or when we talk about the cheetahs with a particular pattern, they are very less number as compared to the other cheetahs which are dominant in a lot of national parks. Even when we come to study of certain deers, so when we are talking about certain kind of deers, the actual population of certain deers has gone down, and that is because of a bottleneck effect, as a result of which the entire gene pool or the entire set of genes which are there in total have been reduced for a particular organism or a particular species, and the other ones have become dominant. So a particular animal gets lost or becomes extinct. So the yellow ones in these case kind of look like they have completely become extinct, and the species fall in number. The whole population does go down. Obviously the whole population is also going down, but that will be recovered over time because the ones which have survived will increase in number. So there is an unlimited talk which can go about bottleneck and founder effect. Coming back to what you need to understand with a reference to the exams, founders effect is something which is asked to you, where they ask you to explain is it happening because there is a reduction in gene pool or not. So please remember, whenever you are answering the questions for founder effect as well as bottleneck effect, try and use the word as gene pools, try and use the word as variations coming into place, try and use the word selection pressure which is coming into place. So when we are talking about bottleneck effect, there is reduction in gene pool, however it does not occur by chance, it is because of a selection pressure. Whereas in case of founders effect, it is by chance and there is no reduction of a particular set of gene in your gene pool, the gene pool remains constant. So the next topic which we move on to is the Hardy-Weinberg law. So when we are talking about the Hardy-Weinberg law, it is just talking about distribution of the dominant and the recessive alleles, distribution of the dominant individuals, which will be homozygous for the dominant alleles, heterozygous for the dominant alleles, and homozygous for the recessive alleles, trying and finding their frequency with respect to the entire population. As when we started with the chapter, we started with topics of variation, then we moved on to selection and how selection comes into play. When we are talking about selection happening for a particular type of allele in a particular environment, we need to put it down mathematically into numbers, and that's where we have this Hardy-Weinberg law coming in, and that's where this question is kind of asked a lot in paper 5, where we have a lot of mathematical calculation coming in. It is also asked in paper 4 as one of the major questions, where they do give you frequencies. So please remember, there are two set of equations in terms of mathematics we are talking about, and I really don't want those students to worry who think that, oh no, why have we got mathematics introduced here. You just need to understand a very basic mathematical level which you had been studying since your middle school years, not very complex mathematics or quadratics which is coming into place here. So the first equation is P plus Q. Now what is P? P is not the dominant allele, but the frequency of the dominant allele. That is, in a particular community or in a particular population setup I have, how much has my dominant allele occurred out of the total number of alleles. So both of the dominant and recessive allele are the two types of allele which will occur, and it is their combinations which will create individuals. So when I talk about these two alleles, both of them make 100%. So the values of P will always be less than 1 and value of Q will always be less than 1. Now by simple mathematics, if you square both the sides, it is P square plus Q square plus 2PQ is equal to 1 square, which is equivalent to 1. And here what I am referring to are individuals. When I was just talking about P, that was just talking about the allele here. However, when I am talking about the offspring, that's when I start talking about P square. So P square is when my offspring is inheriting the dominant allele. Q square is chance of my offspring inheriting the recessive allele, and Y is PQ twice. So here I'm talking about heterozygous individuals, but my heterozygous individuals can have the dominant from the father and the recessive from the mother, or vice versa. So I need to consider both of those chances, and that is why it is 2PQ. And obviously the frequency of all of these will make 100%, and that is 1. So here I am not talking about population numbers. For example, if I say that in my population of about 100 individuals, okay, my uh, individuals which have uh, only the recessive phenotype are 20. Okay, so what is my q square value? Because I know about the individuals, it will be 20 divided by 100. Okay, which is zero point 0.2. So using this, I should be able to calculate my other values very easily. But please remember, when we are talking about numbers here, we are talking about percentages and frequencies. We are not talking about whole numbers. The major mistake which most of the students make is that they don't calculate the frequencies of the allele or the frequencies of the offspring. And how do we calculate? We calculate by the number which is given to you by the total population. Please remember, most of the values in the question paper start from q square, and then you can easily move on to using your calculators, which are allowed during your exams, to do an under root and calculate the values. Okay. um, in my next set of video, I will try and move on to a few sample questions of Hardy-Weinberg law. The topics which are usually left after the Hardy-Weinberg law are basic examples of how selection has introduced evolution, or how it has actually introduced extinction. One last topic which is also left under selection and evolution is the speciation topic, on which we do get a few questions, and that's what I'm going to cover in my next video. Thank you so much, and please put on any questions you have, and I will be happy to answer. Thank you.